Hello, everyone, and welcome back to our series on the hallmarks of cancer. We are on episode 4, which talks about the new and malignant and the immortal daughter cell, which discusses the acquired capability of limitless replicative potential. So, as an analogy for this, imagine you have a clockwork toy. Now, normally, this toy is a spring that winds down, limiting how long it can run before it needs to be wound up again. This winding down process is similar to how our cells have a built-in limit on how many times they can divide before they stop. Now picture a magical clockwork toy that never winds down. Instead of stopping at a certain time, it keeps going endlessly, never needing to be rewound. This endless operation is akin to what happens in cancer cells. They develop the special ability to bypass the normal limits on cell division. This ability is called limitless replicative potential, and just as a magic toy keeps running without stopping, cancer cells can keep dividing indefinitely. Normally, our cells have a kind of internal clock. They're telomeres that shorten every time they divide. Telomeres are protective caps at the ends of our chromosomes composed of repetitive DNA sequences. When the telomeres get too short, the cell stops dividing. However, cancer cells find a way to keep their telomeres from shortening, and this allows them to keep dividing without limits, like an endless clockwork toy. Now let's talk about telomeres. Every time a cell divides, the telomere gets a bit shorter. During each cell division, the telomere shorten due to the inability of DNA polymerases to completely replicate the ends of DNA molecules. This progressive shortening eventually leads to the exposure of chromosomal ends, which causes crisis and a massive cell death due to chromosomal instability. When the telomere gets too short, the cell knows it's time to stop dividing and it enters a state called senescence, which is like a retirement phase for cells. And senescent cells are still metabolically active, but they can't replicate, which prevents the spread of potential DNA errors. If the cell keeps pushing past this point, it enters something known as crisis, where most cells die because their DNA gets messed up, which leads to karyotypic disarray. In crisis, because the telomeres are too short, the ends of chromosomes can fuse together or break apart improperly, which can cause severe chromosomal abnormalities. This disarray often causes cell death because the cell's genetic material becomes too damaged to function properly. But cancer cells can find a way to cheat the system, and they do this by activating an enzyme called telomerase. Think of telomerase as a magic gardener that adds new pieces to the ends of the telomeres, keeping them long. This way, cancer cells can keep dividing without ever hitting that crisis point. Telomerase adds telomeric repeats at the ends of chromosomes, which maintains the length and allows indefinite cell division. This capability is observed in around 85 to 90% of cancers, and by maintaining the telomeres, cancer cells can avoid crisis and continue proliferating. However, I did say 85 to 90, so what about the 10 to 15% that do not use telomerase? Well, they use another mechanism called ALT. ALT stands for alternative lengthening of telomerase. And think of it as like a clever construction workaround. Instead of using new materials like telomerase, these cancer cells use existing parts from other buildings to extend the structures. This is achieved through a process called homologous recombination, which we also observe in meiosis, where telomeres borrow sequences from other chromosomes to patch themselves up. In cells using ALT, this recombination happens in specialized areas called ALT-associated PML bodies, where PML stands for pro. This is very difficult for me to pronounce. That's why I wrote this part down. Promyelocytic leukemia bodies. So these are nucleus structures that have a variety of proteins that are involved in different cellular processes, including tumor suppression, DNA repair, and cellular stress levels. So this probably gives you an indication to how important PML bodies are in cancer. So these are composed of PML proteins and other associated proteins that regulate these activities, and they are involved in cellular growth, apoptosis, and the maintaining of genome stability. Now, a unique marker of ALT activity is the presence of C circles, which are small circular DNA structures made from telomeric sequences. These can be detected in laboratory tests, indicating that a cell is using the ALT pathway. And it's important to know about ALT or alternate mechanisms, especially the alternative lengthening of telomerase, because it allows us to treat a wide variety of cancers in a multi-pronged way, so that they don't evolve different ways and learn to evade the treatment that we already have. Now we're going to be talking about the Hayflick limit. And normal cells have a finite replicative period, meaning that they can divide only a certain number of times before they stop. And this was first observed by the scientist in the 1960s called Leonard Hayflick, and is known as the Hayflick limit. His work showed that cells in a culture have a finite number of divisions, I said 60 to 70, before they enter senescence. However, cancer cells can bypass this by disabling key tumor suppressor proteins like PRB, which is the retinoblastoma, and P53, which allows them to evade senescence and continue dividing. So over here are the key terms if you want to pause and just take a look at what I mentioned. And the reason that it's so important to understand this hallmark is because these mechanisms are crucial in helping researchers develop treatments that can target specific capabilities of cancer cells. And it can potentially limit or control or stop their uncontrolled growth. By targeting telomerase or the ALT pathway, scientists are trying to find ways to limit the replicative potential of cancer cells and halt their progression. And I hope this explanation was helpful, and I hope you understand how cancer cells manage to grow without limits. So yeah, to the two people who are going to watch this video, please like and subscribe for more of these. And I'm looking forward to my first comment. Thank you.